Hello student, good morning. Myself Sri K.D. Borsesar, biology subject teacher. In previous lecture we learned cell structure and organization, chapter content concept, component of eukaryotic cells. You know, in eukaryotic cell consist of three components: cell envelopes, cytoplasm and cell organelles. In previous lecture we discussed cell organelles like mitochondria. You know, mitochondria is one of the most important cell organelle, powerhouse of the eukaryotic cell, because it generate and store ATP energy. Also we learn in previous lecture plastids. You know, plastids are consist of three types: leucoplast, colorless plastid; chromoplast, colored plastid other than green color; and chloroplast is green colored plastid. Chloroplast is involved in process of photosynthesis, production of glucose. Now today we learn some more cell organelles of eukaryotic cell, like endoplasmic reticulum is one of the cell organelle. Golgi complex is one of the cell organelle, while lysosomes are also one of the cell organelles. So let us start today's first concept. You know, eukaryotic cell organelles. Eukaryotic cell organelles consist of endoplasmic reticulum, Golgi complex and lysosome. Today we learn in detail these three cell organelles. In previous lecture we discuss mitochondria as well as plastid. Today we learn these three cell organelles. So let us start one by one. First one is endoplasmic reticulum. In short, ER. E means endoplasmic, R means reticulum. Endoplasmic reticulum is a little network present in the cytoplasm. It is a little network present in the cytoplasm. It present in all eukaryotic cells except OHA means egg cell and mature red blood corpuscle means RBC. Always remember, egg cell or OHA, also mature RBC, absent endoplasmic reticulum. Under electron microscope, endoplasmic reticulum appear network of membranous tubules called as cisternae. Under electron microscope, endoplasmic reticulum appear network of membranous tubule called as cisternae. See near diagrammatic representation. These are all cisternaes. So membranous tubule like cisternae structure seen in electron microscope, endoplasmic reticulum. It form more than 50% of the total membrane of eukaryotic cell due to membranous network tubule of cisternae. Endoplasmic reticulum present in eukaryotic cell form more than 50%. Always remember, 50% or more percent ER are present in eukaryotic cell. Due to ER means endoplasmic reticulum, cytoplasm divide into two parts. Means endoplasmic reticulum present असल्यामुळे सायटोप्लाझमचे दोन पार्ट्स होतात. नंबर 1, within the lumen of ER called as laminar cytoplasm. ERच्या ल्युमेन मध्ये असलेला सायटोप्लाझमचा पार्ट लॅमिनार सायटोप्लाझम, while outside ER called as non-laminar cytoplasm. Means due to ER cytoplasm divide into two parts, laminar cytoplasm, non-laminar cytoplasm. One end of ER connected with nuclear membrane, while other end of ER connected to cell membrane or plasma membrane. Always remember, one end of ER connected to nuclear envelope, while other end of ER connected with cell membrane. Let me show with the help of diagram here. Clear, one end is attached to nuclear membrane. This is the endoplasmic reticulum. This one is also endoplasmic reticulum. So one end of endoplasmic reticulum is attached to cell membrane or plasma membrane. This is the plasma membrane, while other end of endoplasmic reticulum attached to nuclear envelope, both side, right side as well as left side. So this is the most important due to both end attach at different places. Cell support provide endoplasmic reticulum. Does ER acts as intracellular supporting network? Because one end is attached to nuclear membrane while other end is attached to cell membrane. Due to this, ER acts as intracellular supporting network, help maintaining position of the various cell organelles. In the cell cytoplasm, various cell organelles are maintaining in the position or location because of intracellular supporting network of ER. According to present or absent ribosome, on outer surface of endoplasmic reticulum, consists of two types. Number one, rough endoplasmic reticular and smooth endoplasmic reticular. In short, RER, rough endoplasmic reticulum. SER, smooth endoplasmic reticulum. Both type of ER is due to presence or absence of ribosome on outer surface of endoplasmic reticulum. Let me show with the help of diagram. Rough endoplasmic reticulum and smooth endoplasmic reticulum seen here. On outer surface of endoplasmic reticulum present ribosome. These are the ribosome. All these spots are ribosomes. When outer surface of the ER present ribosome, such ER called as rough endoplasmic reticular, while on outer surface, here clearly seen on outer surface of ER, do not show ribosomes. So such ER endoplasmic reticulum called as smooth endoplasmic reticular. Up to this we detail discuss total 7 different characteristic features of endoplasmic reticulum, and on the basis of present or absent of ribosome on outer surface of ER, two types of ER, rough endoplasmic reticular, smooth endoplasmic reticular. Hope that you understood everything. Now we learn function of rough endoplasmic reticular. Number 1, RER means rough endoplasmic reticular is primarily involved in protein synthesis. Which type of ER primarily involved in protein synthesis? Rough endoplasmic reticulum. Example, pancreatic cell that secrete insulin is the best example of protein synthesis. Such protein are secreted by ribosome attached to RER called as secretory protein. Third function, rough endoplasmic reticular also involved in formation of cell membrane. Also involved in formation of cell membrane. Up to this we detail discuss rough endoplasmic reticular function. Now we learn smooth endoplasmic reticular function. Number 1, SER means smooth endoplasmic reticular involved in various processes in different cells. Means वेगवेगळ्या cells मध्ये वेगवेगळ्या process मध्ये involve होतो smooth endoplasmic reticulum in the cells. Number 1, SER helps synthesis of lipid. You know, RER helps synthesis of protein, but SER helps synthesis of lipid. Example, number 1, steroid secreting cells of cortical region of adrenal gland. Cortical region मध्ये adrenal glandच्या cells मध्ये SER असतो आणि तो काय secrete करतो? Steroid secrete करतो, and steroid is also one of the lipid. Also, testes and ovaries contain SER for synthesis of lipid. Always remember, in steroid secreting cells, cortical region of adrenal gland contain SER synthesis lipid. Testes and ovary contain present SER synthesis of lipid. SER play a role detoxification of drugs and poisons in the liver cell. Liver cell present SER play important role detoxification of drugs or poison. Drugs and poison मध्ये असलेलं टॉक्सिक न्यूट्रल करण्याचं काम करतो SER, and such process seen in liver cell. Now, storage of calcium ions is also one of the main function of SER seen in muscle cells. Muscle cell contain SER storage calcium ions. Up to this here, 3 different function noted. SER means smooth endoplasmic reticulum, and up to this we detail discuss endoplasmic reticulum is one of the cell organelle, consist of 2 type RER and SER. Both types of ER provide different function. Hope that you understood everything. Now we learn today's second cell organelle known as Golgi complex. Golgi complex is one of the cell organelle of eukaryotic. Golgi complex, also called as Golgi apparatus or Golgi body, because it first described by Chamelo Golgi So such these organelles are name given to Golgi complex, Golgi apparatus, Golgi body. Golgi complex is assembly, manufacturing, packaging and transporting unit of the cell. Which one is assembly, manufacturing, packaging and transporting unit of cell? Golgi complex. It consists of stalk of membranous sac called as cisternae. Means Golgi complex consists of stalk of membranous sacs called as cisternae. Seen here diagrammatic representation. This is the cisternae, this also cisternae, all these are the cisternaes. Label here cisternae, and this cisternae is look like stalk of membranous sac. Diameter of cisternae varies from 0.5 to 1 micrometer. Here seen, this is the diameter of cisternae. It varies from one another from 0.5 to 1 micrometer. So as cell may have few or several cisternae, means एका सेल मध्ये सिस्टर्नीची संख्या फ्यू काही असू शकतात किंवा सेव्हरल नंबर्स ऑफ असू शकतात. डिपेंड ऑन इट्स फंक्शन. त्यांच्या फंक्शनवर डिपेंड करतं संख्या किती असेल. each and every cell. Up to this we detail discuss. Total 5 characteristic features of Golgi complex. Now point number 6. The Golgi sacs show specific orientation in the cell. Golgi sac show specific orientation in the cell. Each cisternae has a forming cis phase. Each cisterna has a forming cis phase, cis on the same side, and maturing or trans phase. Means जेव्हा सिस्टरनी फॉर्म होते तेव्हा सिस फेज मध्ये ओरिएंटेशन असतं गोल्गी कॉम्प्लेक्सचं. तीच सिस्टरनी जेव्हा मॅच्युरिंग होते तेव्हा ट्रान्स फेज मध्ये ओरिएंटेशन दिसून येतं. कोणाचं? तर सिस्टरनीचं. सो सिस मीन्स सेम साइड, ट्रान्स मीन्स अपोजिट साइड. मीन्स जेव्हा फॉर्मिंग होते तेव्हा सिस फेजमध्ये असते आणि जेव्हा ती मॅच्युअर होते तेव्हा ट्रान्स फेजमध्ये असते. आणि सिस फेज आणि ट्रान्स फेज हे एकमेकांचे काय तर अपोजिट असतील. सीन हिअर दिस इज द सिस फेज. फेस मीन्स फॉर्मिंग cisternae and this one is trans phase. Always remember cis phase and this one is trans phase orientation. When forming of cis phase same side, while maturing of trans phase opposite side. The cisternae of Golgi complex mature moving from cis phase to trans phase. Always remember cisternae of Golgi complex mature moving from cis phase to trans phase. Means this is the cis phase is transfer maturity to trans phase. Now this is called as cisternal maturation model. Means how maturation process takes place. Cisternae of Golgi complex mature moving from cis phase to trans phase. It is called as cisternal maturation model. Hope that you understood everything. What is cisternal maturation model? The cisternae of Golgi body are not physically connected with each other. Here noted, each cistern is separate from one another. Means cisternae of Golgi complex are not connected with each other, and this is also one of the most characteristic feature of Golgi complex. Up to this we detail discuss total 9 characteristic features of Golgi complex. Function of Golgi complex. Number 1, Golgi body carry out 2 types of function. Golgi body carried out 2 types of function. Number 1, modification of secretion of ER. ERचं जे सिक्रेशन असतं त्याला मॉडिफिकेशन करण्याचं काम करतो गोल्गी कॉम्प्लेक्स. सेकंड, प्रोडक्शन ऑफ इट्स ओन सिक्रेशन. स्वतःचंही सिक्रेशन आहे. सो गोल्गी बॉडी कॅरी आउट 2 types of function. Number 2, cisternae contain specific enzymes for specific function. Cisternae of Golgi complex contain specific enzymes for specific functions. Formation of glycolipid and glycoprotein is also one of the most important function of Golgi complex. Golgi body also manufacture their own product. स्वतःचे ओन प्रोडक्ट मॅन्युफॅक्चरिंग करण्याचं काम करते गोल्गी कॉम्प्लेक्स. गोल्गी बॉडी इज इन मेनी प्लांट सेल प्रोड्यूस non-cellulose polysaccharide like pectin. Golgi body in many plant cell produce non-cellulose polysaccharides like pectin. Up to this we detail discuss total 5 different functions of the Golgi complex, and no doubt up to this we detail discuss Golgi complex is one of the cell organelle of eukaryotic cell. Now we learn lysosome is one of the cell organelle. Lysosome is a membrane bound vesicle. Lysosome is a membrane bound vesicles containing hydrolytic enzymes. Means लायसोझोम हा एक व्हेसिकल आहे. कोणत्या प्रकारचा? तर मेम्ब्रेन बाउंड, आणि त्याच्या आतमध्ये काय असणार? तर हायड्रोलायटिक एन्झाईम्स असतील. मोस्ट ऑफ टाइम दिस क्वेश्चन इज नोटेड. इन विच सेल ऑर्गेनेल कंटेन हायड्रोलायटिक एन्झाईम? Lysosomes. Senior, lysosome structure. This is the membrane bound covering and it contain hydrolytic enzymes. It is a bag like or pouch like small body structure present in the eukaryotic cell. It is waste disposal system of the cell. Means it is cell waste disposal system. सेलची ही वेस्ट डिस्पोजल सिस्टीम म्हणून ओळखली जाते, बिकॉज इनसाइड द सेल Dead cells are disposal or damaged cells are disposal by lysosomes. So, lysosomes are called as waste disposal system. Lysosome also called as suicide bag or autophagic vesicle. Why lysosome are called as suicide bag or autophagic vesicle? Because it brings digestion of old, useless or damaged cell organelles in the cell. So, lysosome called as suicide bag or autophagic vesicle. Number 5. Most of eukaryotic cell lysosome contain enzymes used to digest or hydrolyze macromolecules. eukaryotic cell मध्ये असलेले macromolecules डायजेस्ट करून टाकतो कोण तर लायसोझोम. 9. Lysosome contain hydrolytic enzymes like number 1 hydrolysis, second amylases, proteases and lipases. Here noted all these 4 different types of enzymes are present in lysosome: hydrolysis, amylases, proteases and lipases. Lysosome enzyme are activate only when lysosome come in contact with other cell organelles to form hybrid structure. Always remember, this is most important point. Lysosome enzyme present in lysosome activated only when lysosome come in contact with another particular cell organelle to form hybrid structure. Means lysosome मधले एन्झाईम तेव्हाच ॲक्टिव्ह होतात जेव्हा लायसोझोम दुसऱ्या सेल ऑर्गॅनेलच्या कॉन्टॅक्टमध्ये येतो आणि जे स्ट्रक्चर फॉर्म झालेलं असतं ते काय असतं? हायब्रिड स्ट्रक्चर. Lysosomal enzyme do not digest their own membrane protein. स्वतःचा ओन मेम्ब्रेन प्रोटीन कधीही लायसोझोमल एन्झाईम डायजेस्ट करत नाही. Always remember. 9 number point. Remember, every week a human liver cell recycle half of its macromolecules. Remember, every week a human liver cell recycle half of its macromolecules. Up to this we detail discuss total 9 different characteristic features. In that 4 different types of hydrolytic enzymes like hydrolysis, amylases, proteases and lipases. Now, types of lysosome. Number 1, first, primary lysosome. What is primary lysosome? Those lysosome contain hydrolytic enzyme in an inactivate state. Such lysosome called as primary lysosome. Second type of lysosome, secondary lysosome or hybrid lysosome. Secondary lysosome or hybrid lysosome formed by fusion of lysosome with endocytic vesicle containing material to be digested, represented as heterophagic vesicle. Also, this is the larger size than the primary lysosome. Here clearly seen primary lysosome is different from secondary lysosome. Primary lysosome contain hydrolytic enzyme in an inactivate state. Secondary lysosome, also called as hybrid lysosome, because it formed by fusion of lysosome with endocytic vesicle that contain material to be digested, and this is called as heterophagic vesicle. I'll always remember, secondary lysosome is larger size than the primary lysosome. Third type of lysosome called as residual body. A residual body type of lysosome is one of the type of lysosome vesicle containing undigested remains. Such lysosome contain undigested remains, so such lysosome called as residual body. Fourth type of lysosome called as suicide bag or autophagic vesicle, and no doubt suicide bag or autophagic vesicle is digest damaged cell organelle. Here noted total 4 different types of lysosome. Primary lysosome, secondary lysosome also called as hybrid lysosome, a residual body type of lysosome, and suicide bag or autophagic vesicle is also one of the type of lysosome. Up to this we detail discuss different characteristics of lysosome and types of lysosome. Now we learn function of lysosome. Number 1, first function of lysosome. Lysosome digestion of old or useless or damaged or dead cell organelles. Such process called as autophagy. Means ज्या सेल मधला लायसोझोम असेल त्याच सेल मधले ओल्ड युजलेस डॅमेज डेड सेल ऑर्गॅनेल्सला डायजेशन करतो, त्या प्रोसेसला आपण ऑटोफॅगी म्हणू शकतो. सेकंड, डायजेशन ऑफ फॉरेन मटेरियल बाय सेकंडरी लायसोझोम. हेटेरोफॅगी means other than the self material digestion takes place. Such process called as heterophagy. Self material digested called as autophagy. Third function, lysosome bring about intracellular and extracellular digestion. सेलच्या आतमधले मटेरियल डायजेशन करतो, सेलच्या बाहेरचा पार्टही डायजेशन करतो. सो लायसोझोम ब्रिंग अबाउट इंट्रासेल्युलर अँड एक्स्ट्रासेल्युलर डायजेशन. फोर्थ फंक्शन, इंट्रासेल्युलर डायजेशन is brought about by phagocytosis. Means eating dead, damaged, useless cell called as phagocytosis, and this is the best, most important example, intracellular digestion. Fifth function, extracellular digestion is brought about by release of lysosomal enzyme outside the cell. जेव्हा सेलच्या आउटसाइड लायसोझोमल एन्झाईम रिलीज केला जातो तेव्हा एक्स्ट्रा सेल्युलर डायजेशन प्रोसेस होते. एक्झाम्पल, डिझॉल्विंग ओहम मेम्ब्रॅन. ओहम मेम्ब्रॅन मीन्स एग मेम्ब्रॅन डिझॉल्व केला जातो. कोण डिझॉल्व करतो? बाय ह्युमन स्पर्म लायसोझोम ॲक्रोझोमल कॅप. ह्युमन स्पर्मची ॲक्रोझोमल जी कॅप असते त्या कॅपमध्ये लायसोझोम असतो. अँड सच ॲक्रोझोमल कॅप कंटेन लायसोझोम सिक्रेट एन्झाईम हायलुरोनायडेज. सिक्रेट एन्झाईम known as hyaluronidase That enzyme is responsible for dissolving the ovum membrane, bring fertilization. You know fertilization, union of male and female gametes to form zygote, such process called as fertilization. So union of male and female gamete, at that time dissolving the female gamete ovum membrane, is done by lysosome acrosomal cap secreted enzyme hyaluronidase. So bring fertilization process. Hyaluronidase is one of the most important enzyme, are essential during the fertilization because it help dissolving the egg or ovum membrane. Due to this, human sperm enter into the egg or ovum, and due to this fertilization process is completed. Hope that you understood everything. Hyaluronidase is one of the enzymes secreted by acrosomal cap lysosome part of the human sperm. Now we learn function number 6, reusing the tissue of redundant organs during metamorphosis. जे ऑर्गन्स रेडुंडंट झालेले असतात त्या ऑर्गन्समधले टिश्यू रियुजिंग करून मेटामॉर्फोसिस प्रोसेस होते, अँड इट इज डन बाय लायसोझोम. नाउ 7 नंबर फंक्शन, डिस्ट्रक्शन ऑफ malignant cells. Example, T lymphocyte. You know T lymphocyte is one of the most important cells of WBC that destroy malignant cells, means cancerous cells. So these T lymphocyte cells contain lysosome, are useful for destruction process of malignant cells. Up to this we detail discuss total 7 different functions of the lysosome. Up to this we detail discuss lysosome is one of the cell organelle. Review today's topics. We learn in today's lecture eukaryotic cell organelles like endoplasmic reticulum, that is ER, Golgi complex, and lysosomes. On the basis of today's lecture, some exercise question noted here. Number 1, MCQ question. Lysosomes are not helpful in osteogenesis, cellular digestion, metamorphosis, lipogenesis. Yes, the correct answer is lipogenesis, because lysosome is not involved in lipogenesis process. Second, Golgi body is absent in prokaryote, mature mammalian RBC, OHA, all the above. Yes, the correct answer is all the above, D option. Next, number 3rd, dash is involved in detoxification of the liver cell, RER, Golgi bodies, SCR, mitochondria. Yes, the correct answer is SCR, smooth endoplasmic reticulum, involved in detoxification of liver cell. Now, assembly, manufacturing, some packaging and transport unit of the cell is ribosome, Golgi body, SCR, mitochondria. Yes, the correct answer is Golgi body, is useful for assembly, manufacturing, some packaging and transport unit of the cell. Short answer question number 1. Which are the two types of ER? Yes, RER, rough endoplasmic reticular, and SER, smooth endoplasmic reticular. Second, which cell organelle called as suicide bag? Why? Yes, lysosome cell organelle called as suicide bag because lysosome digest old cell organelles, useless cell organelles, and damaged cell organelles in the cell, so lysosome called as suicide bag. Lysosome bring about dash dash and dash dash digestion. Lysosome bring about intracellular digestion and extracellular digestion. Fourth, enlist lysosome contain hydrolytic enzymes. Yes, number 1, hydrolysis. Second, amylases, proteoses, lipases. Next, number 5, which type of ER primarily involved in protein synthesis? Rough endoplasmic reticulum involved in primarily protein synthesis process. Function of SER, synthesis of lipid is the main function of SER. Also detoxification of material in the liver. Dissolving Ohm membrane by human sperm. Lysosome acrosomal cap secrete enzyme that responsible for or bring fertilization. Yes, hyaluronidase enzyme secreted by acrosomal cap lysosome of human sperm. Difference between RER and SER. Yes, endoplasmic reticulum on outer surface present ribosome. Such endoplasmic reticulum called as rough endoplasmic reticular, while smooth endoplasmic reticular means on ER means endoplasmic reticular outer surface ribosomes are absent. Such ER called as smooth endoplasmic reticular. Identify the cell organelles or its parts from its description below. खाली दिलेल्या डिस्क्रिप्शन नुसार सेल ऑर्गॅनेल्स ओळखा किंवा त्याचे पार्ट्स ओळखा. नंबर 1, इन टेस्टिस अँड ओव्हरी सिंथेसिस ऑफ लिपिड. यस, which cell organelle in testes and ovary synthesis of lipid? SER means smooth endoplasmic reticulum is a part of endoplasmic reticulum cell organelle. Digest damaged cell organelles. Yes, lysosome digest damaged cell organelle. Third, acts as intracellular supporting network. Yes, endoplasmic reticulum acts as intracellular supporting network. Formation of cell membrane is the most important function of rough endoplasmic reticulum. Identify the given diagram and labeled A and B. Yes, this is the endoplasmic reticulum diagram. Yes, endoplasmic reticulum, on outer surface present ribosome. These are the ribosomes. So such endoplasmic reticulum called as rough endoplasmic reticular, means A means rough endoplasmic reticular. Yes, this is the B. On outer surface of endoplasmic reticular not present ribosomes, so such ribosome not present ribosomes, so such endoplasmic reticular called as smooth endoplasmic reticular, means B means smooth endoplasmic reticular, A rough endoplasmic reticular, and the given diagram known as endoplasmic reticulum. Up to this, today's lecture completed. Hope that you understood everything. If you like the video, then you can subscribe my YouTube bioinfo channel. Tap the bell icon and like the video. If any doubt about today's lecture, mention in comment box. Thank you.